The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Now, before we get started, let me introduce our speaker. Dr. Richard Frye is a child neurologist and director of autism research at Arkansas Children's Hospital Research Institute. He's an associate professor in pediatrics at the University of Arkansas for Medical Sciences and director of the Autism Multispecialty Clinic at Arkansas Children's Hospital. These webinars are made possible through generous donor support. If you'd like to contribute, please visit our website at autism.com. And now I'll turn this over to Dr. Frye.
Well, hello. Thank you so much for inviting me to talk about folate metabolism, mitochondrial disease, and how it relates to autism today. Some of my favorite topics, and I'm glad there's so much interest in knowing more about this. So again, thanks for this opportunity, and I will start the presentation. As many of you know, autism is somewhat of a mystery, and the reasons for developing autism is not quite clear. One of the things that we like to concentrate on at my center is the metabolic abnormalities that are associated with autism, particularly those that we can treat. That is that it's important, of course, to look for all types of disorders that may be associated with autism, but I think some of the most important things to look for are things that you can do something about to improve the lives of individuals with autism. Today we'll concentrate on the mitochondrial disorders and disorders of folate metabolism. And what's interesting is that in autism we find that there's several different types of mitochondrial disorders. That is, there's what we call classically defined mitochondrial disease, and that's what widely people think of in terms of mitochondrial disorders. However, we believe there's more of an expanding range of mitochondrial disorders where the mitochondrial diseases, as they're strictly defined, is just one example. And in autism, we find that about 5% of children can be diagnosed probably with a mitochondrial disease. Whereas we also see some other very novel changes in mitochondrial function, which appear to suggest that the mitochondria is not working normally, although they may not fit into this classic definition of mitochondrial disease. Particularly, some of the things that we've found is that we find the electron transport chain, that common pathway that generates energy in mitochondria, instead of being underactive, as you see in many mitochondrial diseases, it's actually overactive for some reason, and we think it has consequences when it's very overactive. We've also found that there are these unique abnormalities in fatty acid oxidation metabolism, which we think may be related to the microbiome, although this is kind of preliminary. The other area that's really been very fruitful as far as treatment is treatment of folate abnormalities, particularly abnormalities in central folate metabolism and central folate insufficiency. That is, that it appears that the central nervous system may not have enough folate to really function up to its optimal capacity. And this, we believe, for many children is due to these autoantibodies that affect the transporter that transports folate into the nervous system, the so-called folate receptor alpha. We know that folate's not only important as a vitamin, but it's involved in many pathways that are also found to be abnormal in children with autism. Particularly, it's important for the production of glutathione, the major antioxidant of the body, which is found to be low in children with autism. It's very important for DNA and RNA production, so it's important when cells divide, when they replicate, when they repair those cells. It's important for neurotransmitter production and for mitochondrial function also. And I'll show you some data that we recently found that we thought was actually kind of surprising to show that folate seemed to be very helpful for those with mitochondrial dysfunction. So first we'll talk about mitochondrial disorders and its treatments. The mitochondria is a subcellular organelle. It is located in every almost every cell of your body, and then you don't just have one. You actually have somewhere between 100 and tens of thousands of mitochondria per cell. Mitochondria is best known for producing energy. So many times the number of mitochondria in the cell is proportional to how much energy is needed by that cell. So things like skin cells may only have a few mitochondria, whereas other cells like muscle cells may have lots and lots of mitochondria. The mitochondria itself is very complex, and we can see that on this picture in the upper part of the panel is a diagram of what the inside of a mitochondria looks like. It has an outer membrane and an inner membrane, and in this inner membrane it has a lot of machinery to help it work. In particular, what's very unique about the mitochondria is that it has its own DNA. So every mitochondria actually has its own instructions for a portion of the machinery that's inside the mitochondria. Most of the DNA that controls mitochondria is in the nucleus of the cell, but some of the genes are actually carried inside the mitochondria. And this has some significance, including the fact that it makes looking at inheritance patterns somewhat complex, because your mitochondria are inherited only from your mother, as opposed to your chromosomes, where you have half from your mother and half from your father. What we also find is that there can be some changes in the mitochondria DNA, which may be just fine when you have certain genes in the nucleus, but when you mix up those genes, as you would when you have a child, sometimes the genes in the mitochondria may not play as well as they should with the genes in the nucleus. So there can be some very complex inheritance patterns. Mitochondrial disease is really a new field in medicine. Really, the first mitochondrial diseases were described in 1988 in two papers that were published in Science and Nature, where they found mutations in mitochondrial DNA that were associated with the mitochondria not working correctly. 1988 really is a blink of the eye in the time scale of medicine. Things take a long time to change in medicine and a long time to be understood. So we can understand why mitochondrial disease is something that isn't all that well understood at this point. As you would expect, those that have mitochondrial disease have symptoms in tissues of the body, organs of the body that are very highly energy dependent, like the nervous system, the GI system, and the immune system. And interestingly, these are systems that we see dysfunctional in children with autism. The mitochondria is not just the powerhouse of the cell, but it's also important for regulating redox, that is reactive oxygen species, and it's also key in something called apoptosis, or cell death. Back in 2012, Dr. Rosendahl and I published a paper where we did a meta-analysis and looked at all the evidence for there being signs of mitochondrial disease in individuals with autism. And what we found is that there was 3 studies that looked for the prevalence of classically defined mitochondrial disease in autism. And if you combine those studies, it appeared that the estimate was about 5% of children with autism probably have mitochondrial disease in a very classical sense. However, when we look at markers of mitochondrial dysfunction, that is elevations in lactate, pyruvate, or alanine, or lactate to pyruvate ratio, or low carnitine, we find that those prevalences are much, much higher than the prevalence of mitochondrial disease. And so that made us scratch our head, suggesting that the mitochondria itself may not be working in a larger percentage of children with autism, even though they may not fit the mold of classically defined mitochondrial disease. We also compared the characteristics of children with autism and mitochondrial disease to that of the general ASD population to see if there were any differences. And there were some very interesting differences. We found that children with mitochondrial disease and autism were more likely to have seizures, GI abnormalities, and motor delays as compared to those in the general autism category. And this is important, particularly the fact that we found more motor delays, which is a big red flag, because while many children with autism will have hypotonia, and you can see that hypotonia was the same prevalence in children with general ASD and those with mitochondrial disease, those that had motor delays from early on was much higher in children with mitochondrial disease and autism. So we think of that as a big red flag for children that come in with autism that may have motor delays, which tends to be uncommon in autism. So that's a red flag for mitochondrial disease. The other thing that was surprising is that when we looked at the number of cases where some type of genetic abnormality could account for the mitochondrial disease, we found that only 23% of the cases were there some type of mitochondrial DNA or other type of DNA abnormality or genetic abnormality to explain the mitochondrial disease, suggesting that the mitochondrial disease may not be purely simply genetic, but may be a combination of very complex genetic backgrounds along with environmental influences, or could be polygenetic in a way that's so complex we don't understand it. The prevalence of mitochondrial dysfunction has been somewhat debated in the literature, with estimates that vary widely. This study from the Mind Institute suggested that about 80% of the samples that they looked at, they found that the lymphocytes actually had lower than normal electron transport chain function in the cells they got from children with autism, suggesting that it was very high, the prevalence of mitochondrial dysfunction, at least in immune cells. And they had a follow-up paper with different immune cells where they actually verified this percentage. So they suggest that that this rate may be very high. And what we find also is that there may be these novel types of mitochondrial dysfunction in autism. And this was really a study that is very interesting. I think early on has gone unnoticed, but really I think is probably the first paper to point out some of the subtleties or the uniqueness of mitochondrial dysfunction in children with autism. And in this study, this was a case report of a girl that had something called Leigh syndrome, which is a mitochondrial disease. She did not have autism, and you can see there she's what would be under the column of number 3, case number 3. And when they looked at how her mitochondria would function, they found that complex 4 and complex 5 were functioning below normal. Then they looked at her brother who had autism, and he had the exact same mitochondrial mutations in his mitochondria, which in her caused Leigh syndrome, which is a classic mitochondrial disease. But for him, instead of causing a decrease in function of the mitochondria in complex 4 and 5, it actually caused an increase in complex 1, and complex 1 was functioning much higher than normal. He had fewer mutations, that is, that you have many copies of mitochondrial DNA. And sometimes if you have a mutation, they may not affect all of the copies. Whereas a sister, you can see that about 80% of her mitochondrial DNA were affected. Only 60% of his DNA were affected. So the mutation affected him slightly less. But for some reason, instead of the mitochondria reacting so it wasn't working as hard as it should, that is having a decreased activity, he actually had an increase in complex 1 activity, and he had autism. This idea that the mitochondria might be working harder than normal or be more active has also been shown in complex 4 in brain samples in this study. We published a paper back in 2011 where we had a case series of 5 children with overactivity in complex 4 who had autism, rather low functioning, and many of them had cerebral folate deficiency also. So what does that mean, and what is the significance of that? So one of the things that we've done in our laboratory is we've looked at a model of mitochondrial dysfunction and through using lymphoblastic cell lines, some derived from children with autism, and they were paired to unaffected control children. We asked the question, well, how is the mitochondria working in these cell lines? We use something called the seahorse, and the seahorse is a device that really has advanced our ability to look at mitochondrial function in the sense that it used to be that you could only look at one sample at a time, and you had to isolate mitochondria from cells to examine them. The seahorse is a machine that allows us to look at 96 samples at the same time and look at live cells without having to extract the mitochondria. So we can do all types of experiments with it, and we can put these cell lines in there. It gives us these different measures, which I won't go into in detail, of how the mitochondria is working, including basal respiration, something called ATP-linked respiration, that is how much ATP, which is the energy molecule of the cell that the mitochondria is making, something called proton leak, which tells us about oxidative stress inside the mitochondria. We also look at something called maximal respiratory capacity, which tells us how hard the mitochondria can work if it is driven to its maximum. And we can calculate this thing that's the difference between maximal respiratory capacity and basal respiration, called reserve capacity. And that tells you how much reserve the mitochondria had if it was put under stress. And this is thought to be a very important number because if your reserve is 0, that means that you are working at baseline at your maximal capacity. And anything, any little bit would tip you over the edge to become severely dysfunctional. And studies have shown that if you just deplete your reserve capacity, there's bad things that happen to the cell, such as pathways that induce cell death. So we used this setup to look at these different cell lines, and what you can see in this graph is 4 of these different parameters of how the mitochondria is working. The blue line are the control cell lines, and the red line are the autism cell lines. Then we use something called DMNQ, which you can see on the x-axis, and that is something that stresses the cell. So what we wanted to do is see not only how the cells were working just at baseline, but if we stress them, what would happen to them. So one of the first things that we found, which were somewhat surprising, is that at baseline, and that would be at DMNQ0, we see that the autistic cell lines actually are making more ATP or more energy than the control cell lines. When we look at their maximal respiratory capacity, we see that they're maximal respiratory capacity is actually increased compared to the control cell line. When we look at the reserve capacity, we see that the reserve capacity is actually increased or higher than the control cell lines. And then what we see is that when we stress the cell, they react very differently. That is, the autistic cell lines will lose their reserve capacity faster, and it will go below the control cell lines. So we found this interesting, but of course we look at these differences in the reserve capacity, and they look rather modest. So like a lot of things in autism, we think about subgroups. And what we did is we said, well, maybe there's a subgroup of these cell lines that seem to have this unique ability to function much higher, or have make more ATP and be more overactive than the control cell lines, and maybe there's some that are just like the control cell lines. And so we did something called a cluster analysis, which looked at the difference between the reserve capacity and the autistic cell lines and the control cell lines, and we indeed found that there was two different subsets. One that was just like the control cell lines, and one that was much different than the control cell lines. And then when we graph them out, we can see that the differences were much more dramatic. That is, the top row, we see that the control cell lines and the cell lines labeled AD-N for normal, that they're functioning like the control cell lines, seem to have little difference than the controls, whereas those that are functioning abnormal, called ADAs, which is the second row here, we can see that they're very different than the control cell line. They make about twice as much energy. Their maximal respiratory capacity at baseline is twice that of the control cell lines, and the reserve capacity is almost twice that of the control cell lines. And when you stress them, you see a much more dramatic change with the reduction in reserve capacity going down much faster than the control cell lines. And we think this is important because stressing these cells with increasing reactive oxygen species, we believe, is a good model to look at environmental stresses, since many environmental stresses seem to cause their effect through reactive oxygen species. So this is we think is a model of where you would have some predisposing factor, that is, for some reason the cell lines functioning much higher than normal, and as soon as you put some type of stressor, it actually causes the cell to become dysfunctional and lose its reserve capacity. And we talk about a model of this that is having these two cell lines, one with a maladaptive response and one with a normal response to oxidative stress. And those with a maladaptive response are much more sensitive to stressors. We have also seen that there's other types of unique disorders related to the mitochondria. And the first time I found this, I did a review of some of the patients that went through my clinic to see if there is any common abnormalities that they had. And what we found is that many of them had abnormalities in something called acyl-carnitines, which tells us about fatty acid oxidation. What we found is that when we looked at where these abnormalities were, they consistently had elevations in something called short chain and long chain fatty acids, or acyl-carnitines, without particularly elevations in the medium chain. And really, this is not a disorder that's been described before, because usually fatty acid oxidation disorders have a different pattern, and you would expect that the medium chain acyl-carnitines would be elevated, which you don't see. when I presented this, actually, I just happened to be at a think tank, an ARI think tank that Dr. Derek McFabe was also presenting his work on his propionic acid model, rat model of autism, where he shows that propionic acid can cause autistic-like behaviors. And what he found is that when these rats were exposed to propionic acid, and he looked at their tissue and looked at their acyl-carnitines, the same type of measurement that we do in patients, that he found this exact same pattern of elevations in short and long chain acyl-carnitines, but not medium chain acyl-carnitines. When these patients had been worked up, some of them, because they had signs of mitochondrial disease, did go on to have muscle biopsies. And what we found is that there was a decrease in complex 1 of the electron transport chain, and they didn't have any genetic disorders to explain the problems with complex 1. And what we've published this, and what we found is that if you look theoretically at what a high amount of propionic acid would do to the citric acid cycle, which is in the mitochondria, we would find that actually it would short-circuit some of the key steps in the citric acid cycle, and some of those key steps actually make the precursors to the energy molecules that drive complex 1. And so it would make sense that if you were short-circuiting the citric acid cycle, that complex 1 would be reduced. So we think that this may be, some of these patients may have some mitochondrial dysfunction due to high levels of propionic acid, possibly from microbiome disruptions. How do you treat mitochondrial disorders? Well, there's been no real studies on the treatment of mitochondrial disease in autism, but when we have patients with mitochondrial disease, there's a certain, some people call it the cocktail, to treat children. There has been no studies, again, that have treated mitochondrial disease, but there's been a couple of studies that have used supplements that that would be used in mitochondrial disease in general. And one of the studies from Jim Adams, he put together a very nice supplement that included many of the B vitamins that we use to treat mitochondrial disorders, CoQ10 and acetylcysteine, carnitine, all very important vitamins to treat mitochondrial disorders. And what he found is that when he looked at changes in biomarkers of energy metabolism in children with autism, he found that many of these biomarkers showed improvements when the supplement was taken in the children that took the supplement as compared to a control group. And he also found that compared to individuals taking a placebo, that those that took this simple supplement that could target improvement in mitochondrial function showed improvements in receptive language, hyperactivity, tantruming, and overall functioning. Then there's been 2 studies in something called carnitine. Carnitine has been shown to be low in general in children with autism. For some reason, there's a genetic disorder that's been linked to carnitine metabolism in autism. Carnitine is a very important vitamin. Its major role is to bring fatty acids into the mitochondria to break them down, but it also is very important for eliminating drugs from the body. And there's also studies that show that it's actually neuroprotective for the brain. It protects cells in the brain against oxidative injury. It influences neuronal survival. It's been shown to accelerate regeneration of neurons. And it's also important for mitochondrial function and improves and protects the mitochondria. There's been two studies, one that both of them were double-blind placebo-controlled trials that have used carnitine. In this study, the outcome measure, they used the CARS, the CGI, and then the ATEC. And they found that compared to placebo, those that were on carnitine showed improvements in the CARS score, in the CGI or Clinical Global Impression Scale, and the cognitive subscale of the ATEC. What they also showed is that there was a correlation between how much carnitine improved and how much improvement there was in some of these scores. And there's been a second trial out of Egypt, which wasn't as complex, that also used the CARS, double-blind placebo-controlled study that showed that carnitine was helpful in reducing autism symptoms. So again, these were not children with autism and mitochondrial disease, but in general, this would be a treatment for those with mitochondrial disease, and carnitine seemed to be helpful. So now we'll turn and we'll talk a little bit about folate metabolism, its abnormalities, and treatment. So folate is extremely important. We think of folic acid, which is fortified in our food. It's important to be taken in pregnancy. Folic acid is the oxidized form of folate that isn't completely readily available to the body, which has to be reduced to go into the folate cycle. Other forms of folate, like folinic acid, which you can see in green here, can get directly into the folate cycle. Folate cycle is very important. It supports other areas of metabolism. Folates are the precursors to purines. In particular, purines and pyrimidines are the building blocks of DNA and RNA, which of course is important for gene replication, chromosomal integrity, also the production of certain enzymes, of the machinery of the cell, and also for regulating some of the molecular mechanisms in the cell. So having abnormal folate metabolism can cause problems in DNA replication and DNA repair, which is very important, because your DNA controls your cells and has the instructions for your cells. Also, one of the purines makes GTP, and GTP is a precursor to something called BH4, or tetrahydrobiopterin. Tetrahydrobiopterin is a very important cofactor for making certain neurotransmitters, particularly the monoamine neurotransmitters, and this would include serotonin, dopamine, norepinephrine, and also melatonin. It's also very important for nitric oxide metabolism, which is important for regulating blood flow, and phenylalanine metabolism. On the other side of the folate cycle, we have methylation metabolism. So folate's very important for maintaining proper levels of methyl donors, and without proper folate levels, we have abnormalities in methylation. Methylation, of course, is extremely important for regulation of genes, turning them on and turning them off, and also enzymes, turning them on and turning them off. One of the products of the methylation metabolism is the precursor to making glutathione. And glutathione is our major antioxidant in our cells. It also is important for redox regulation. It can control certain enzyme function. And glutathione is very unique because it's not just important for eliminating reactive species, but it also is a way of getting rid of many toxicants that are in the body, particularly heavy metals. And that actually depletes glutathione. So more has to be made by the body, and it can't simply be recycled. Dr. Jill James did a very important study many years ago where she looked at many of the genes that are important for the folate cycle and its associated, and these also associated pathways, and found that several of the changes in several of the genes that are important for these enzymes will increase the risk of developing autism. And what's really important here is when she actually looked at these changes in these genes and the polymorphisms in these genes, which are common changes that we see, she found that it wasn't just changes in one gene or another gene that increased the risk, but in their analysis they actually found that there were complex interactions. That is, your risk of developing autism wasn't just from having a change in just one gene, but it was when you had combinations. So complex combinations of genetic changes put you at risk for developing autism. Again, this was not finding that these were absolutely deterministic of causing autism, but they put you at risk. So it really suggests that there's many ways of causing inefficiencies in the folate cycle that is a predisposing factor that can lead to autism. One of the things that we study and we think that is very important is folate transport. And it ends up that folate, to get into different parts of the body, needs to go through certain transporters. Also, folate, when you take it orally, is actually transposed into its different forms by the microbiome. The microbiome has can have a very big influence on what is bioavailable if you take folates orally. And there's many different transporters that bring folate into the body, and particularly transporters that take folate and bring it from the blood into the brain. There's two transporters particularly that take folate from the blood and bring it into the brain. One of them is called the folate receptor alpha, and the other one's called the reduced folate carrier. And the folate receptor alpha is very important because it's the main transporter. It has a high affinity for folate, and it's dependent on energy. And the reason why it's dependent on energy is that the levels of folate in the brain are higher than those in the blood. So folate has to be, if you will, pulled from the blood into the brain to have appropriate levels. There's also something called the reduced folate carrier. It has a low affinity for folates. So the same levels of folate in the blood may not bind to it, and it may not transport folate to the same extent as the as the folate receptor alpha. But we found out there's two ways of messing up the folate receptor alpha. One is if you have mitochondrial problems, the folate receptor alpha won't work. And the other one is if you have antibodies that block its function, it will block the ability of the folate receptor alpha to work. What was discovered approximately a decade ago is that there's two different antibodies that can bind to the folate receptor alpha. There's one called the blocking antibody, which actually blocks folate from binding to the folate receptor alpha, and there's the binding antibody, which actually binds to the folate receptor and results in dysfunction because it is bound to it. Folate can get into the brain through an alternative pathway called this reduced folate carrier. About 10 years ago, there was an article published in the New England Journal of Medicine describing a brand new disorder called cerebral folate deficiency. And in this disorder, it was found that children would have regression and other neurological problems starting within the first year of life. What was found also in the cases that were described initially is that many of them had autism. So one of the things we did is myself and a colleague, Dr. Rossignol, looked at children with autism to see if they may have some of these antibodies that would block the ability of folate to get into the nervous system. And what we found is that from the patients we looked at 93 patients, that approximately 60% of them had a blocking autoantibody, either low, medium, or high, and about 50% of them had a binding antibody, either low or medium. That is that about 50% of them were positive for one of these antibodies, and about 25% were positive for both of these antibodies. Children that were positive for these antibodies, we offered them treatment with something called folinic acid, which is a special type of folate that at high doses can get into the brain through the reduced folate carrier, as was shown in many studies on cerebral folate deficiency. And what we found is that when we compared the children that was treated compared to those that were on a wait list, that is that they had gotten the blood test, but they did not know the results, they were not being treated, they had not made any other changes in treatment, we found that children that were treated with folinic acid had significant improvements in receptive language, expressive language, verbal communication, stereotype behavior, and attention. So this drove us to do a double-blind placebo-controlled study, since the first study was open label and there could be bias. We did a study where half of the patients were given placebo, half of them were given high-dose folinic acid to see if the treatment was helpful. And what we found, our primary outcome measure was verbal communication, since we had seen that that was the thing that seemed to improve in our first study to a common extent in children with autism. What we found is that those that were taking folinic acid had a significant improvement in verbal communication in 12 weeks as compared to those that had a placebo. When we looked to see, so one of the things that was very important was to see if this folate autoantibody to the folate receptor alpha had a ability to predict a response. And what we found is that, yes, indeed, those that had the antibody were much more likely to show response to folinic acid. And the number needed to treat for those that were positive for the autoantibody was 1.8, which is a pretty outstanding number for clinical trials. And that would mean that if you have the autoantibody, that more than 1 in 2 will respond significantly in 12 weeks with an improvement in verbal communication. We also calculated how much cost it would be equivalent to for speech therapy, as we were able to see how the improvement in verbal communication was related to the number of hours of speech therapy on an individual basis. And what we found is that a 12-week treatment of folinic acid was associated with probably about a little over $7,000 in speech therapy. And since folinic acid cost about $100 a month, you can calculate the savings would be somewhat substantial. And that's not to say that speech therapy should be stopped, but it can show that the two together may be very helpful. So we think of this as an example of translational research. That is that really this started from observations of children with autism having these folate problems very early on, about a little over 10 years ago, the development of biomarkers and other case series of treated patients, and then doing successively more clinical trials to prove that this therapy is helpful. And I wanted to share with you a recent paper that we published on the effect of mitochondrial supplementations on mitochondrial activity in children with autism. In this study, we actually measured mitochondrial function using buccal swabs at different points. And we knew that some children were on certain supplementations, some were off, and some had held their supplementations to see the effect of the supplementation or how their mitochondria were working without supplementation. We divided the different supplements into these groups, which are the common supplements given for mitochondrial dysfunction. And one of the things we found, which was somewhat surprising, is when we looked at how parts of the mitochondria were coupled, how they were correlated with each other, particularly complex 1 and complex 4 activity, we found that they appeared to be much strongly coupled when somebody was on folate as opposed to when they were not on folate. And when we looked at the coupling of citrate synthase to a complex 1 activity, we found that actually folate and B12 were the two supplements that seemed to significantly improve the coupling of these two enzymes in the mitochondria. And so we believe that folate particularly is very important for mitochondrial function and those with mitochondrial dysfunction. And this makes sense. And one of the reasons it makes sense is, if you remember, I told you that each mitochondria has DNA in it. And one of the ways that the mitochondria compensate when they are dysfunctional is they make more of themselves. And they can't make more of themselves if they can't make more copies of DNA. And if they make more copies of DNA and they don't have the appropriate building blocks like folate, they can actually make errors and actually can induce different types of mitochondrial mutations. So we believe this explains why folate may be very important for those with mitochondrial issues also.
So a lot of the questions that we received were about testing. So if people are interested in getting antibody testing, or if they're interested in delving more into that, can you give any information about how they would get that done?
Sure. Well, there's really the place to get it done is the company's called Iliad Neurosciences, which is in Plymouth Meeting, Pennsylvania. I have to disclose that I am on the scientific advisory board of that company. They have licensed the technology from Dr. Quatros, who was the one that really discovered this antibody. He used to do it on a research basis in his laboratory, but because of kind of the overwhelming request and also the medical implications of it, it really, and now it's more at a company that can do it in a clinical setting as opposed to a research setting. So that can be ordered by any doctor. I don't know that they've worked out whether insurance covers it or not. That's something you'd have to talk to them about. But it costs about $200, and it's something that can very much help decide whether folinic acid would be something that may benefit a particular person.
All right, people are asking for some clarification about L-carnitine or acetyl L-carnitine. So if they're working with supplementation, how do they decide which one to pick?
So really biochemically, you know, the acetyl L-carnitine is broken down into L-carnitine for the most part. So there's really, once it gets into the body, there's not a lot of difference. But there's studies that have used both, so it is very confusing. Many of the studies that have looked at neuroprotection have used acetyl L-carnitine as opposed to just L-carnitine. So in a purest sense, one might say, well, that could be better, but we really don't know. They should be equivalent. But usually what I suggest is to find a carnitine that's tolerated well by the person that's taking it. You know, many supplements have different additives in it or are produced in different ways, so they're not always tolerated all that well people or children. So I would say that there isn't a whole lot of difference, and I would go with the one that seems to be tolerated better.
All right, the next question, I actually received quite a few about methyl B12 and B12 shots. I know you can't really give medical advice, but could you address a little bit about its use with mitochondrial disorders and if it's relevant?
Right. You know, we definitely, one of the things that we think is a standard treatment for mitochondrial disorders, because it's pretty well thought that those individuals that have mitochondrial dysfunction or disease have high levels of oxidative stress. So antioxidants are a mainstay in the treatment of mitochondrial dysfunction. We know from the studies from Jill James that methyl B12 3 times a week subcutaneously injected along with daily folinic acid, low-dose folinic acid, will improve glutathione levels. And glutathione, of course, is very important because it's not only the major antioxidant in your body, but it's a major antioxidant for the mitochondria. And the mitochondria can't make its own glutathione. The cell has to make it for it and import it into the mitochondria. So it's very important to have high levels of glutathione. So we do believe that part of the treatment for mitochondrial dysfunction our methyl B12 shots is a very good treatment, and we see good success in our patients.
So another question I received is about using over-the-counter folinic acid versus using prescribed. So is there a big difference? Is there a benefit to using stuff you can pick up at super supplements, or how can you describe that a little bit?
Well, it's kind of a practical thing. First of all, the over-the-counter folinic acid comes in, I believe, 400 and 800 micrograms. So that's less than a milligram. And when we treat children for problems with central folate issues, we treat them with 25 milligrams twice a day. So essentially, you would have to take a whole bottle of those supplements every day. Not only, financially, probably it wouldn't be the best thing, but all the fillers that they put into the pills probably would hurt your stomach. So I usually suggest that if you're going to use high-dose folinic acid, is what we use to treat problems with central folate problems, use prescription. And I think it's important to be under the care of a medical provider if you're going to do that. For the most part, folinic acid, leucovorin calcium, as it's also known, is very well tolerated. But for any medication that's in that range, it's important to work with somebody that has a lot of experience in treating these disorders.
So here's an interesting question. This person's asking for a little more clarification about the cause of high propionic acid. So that was the McFabe study that you were talking about.
Right. So of course, one of the things that is the theory is that it may be from the microbiome. So we know that organisms like Clostridia, which are propionate producers, are overrepresented in the microbiome of children with autism. So we think that it could be coming from specific bacteria in the microbiome. It is also used as a preservative in certain foods, and it's also in certain grains as a preservative. So it's also possible that we get some from our diet too.
Okay. One person's asking about methyl tetrahydrofolate and its role in treatment.
Yes. And so, what kind of theoretically, it seems that almost any type of reduced folates should be very useful. We use folinic acid because we have the scientific studies that have shown that using it at certain doses will get into the nervous system. Now, it's very possible that other types of reduced folate will do just as good of a job, but we don't have that data right now. One of the other things is that we know that folinic acid, there's a study that suggests that much of it, maybe up to 80% of it, is turned into methylfolate by the microbiome, or it's actually absorbed. So you may be getting best of both worlds by taking folinic acid. You may be getting some methylfolate and also some folinic acid. The folinic acid is thought to also have a regulatory effect on the folate cycle too.